So we are continuing with the second topic, which summarizes what we have learned in epigenetics so far and epigenomics, and that is X chromosome inactivation. Just like imprinting, the X chromosome inactivation utilizes several epigenetic mechanisms at the same time, and therefore it's a good example to review what we have learned so far. So if we read this definition, it says X chromosome inactivation on the bottom, XIC, is both the initial transition from a transcriptionally active to an inactive state of the X chromosome, and also the subsequent stable maintenance of the silent state. So there are two mechanisms in play here. One is the initial transition from an active to an inactive chromosome, and the second one is the maintenance of the inactive chromosome. These are slightly different mechanisms, and therefore it says here the X chromosome inactivation pertains to both of these. So we are going to utilize DNA methylation, nucleosome modifications, interaction with non-histone proteins, interaction of genes with RNA, and changes in nuclear landscape when we cover X chromosome inactivation. All right, this is just a definition, and generally XI means inactive X chromosome, XA means active chromosome. So what is X chromosome inactivation? You might know this already very well. In mammals, we have two X chromosomes in females and one in males, and we inactivate, in other words, silence one of the X chromosomes, and this is primarily because of concerns about dosage. So we are trying to compensate the transcript amounts coming from one chromosome in both genders, because otherwise, if we had allowed both X chromosomes to be active in the females, there are many genes here. Most of those genes would be transcribed at twice the rate at which they are transcribed in males, and apparently this is not what we want. So in mammals, one of the X chromosomes is epigenetically inactivated. But this is not the case in some other organisms. For example, in Drosophila, the rate of expression of genes in chromosome X in males is doubled to compensate for the dosage, whereas in C. elegans, both of the X chromosomes in the female transcriptionally are reduced. So these are, of course, interesting statements, and you might want to find out later on, if you're interested, why people conclude that this is the case. But nevertheless, this is how the situation is. We are going to focus only in mammalian X chromosome inactivation events. So we said X chromosome inactivation is both the initial silencing of an active chromosome as well as the maintenance. The reason why we said those things is because they differ in the mechanism. So you all know that X chromosome inactivation is related to a large RNA transcript, which is called X-cyst. This X-cyst is transcribed from the X inactivation center, which is a region in the X chromosome. When it is transcribed, it acts in cis. It binds to the X chromosome from which it has been transcribed, but it doesn't float around and go and bind the other X chromosome. If the X chromosome center is active, it is going to produce X-cyst, and X-cyst is going to cover the chromosome which is going to become inactive, but it's not going to affect the other X chromosome. The initial silencing is because polycomb repressive complex 1 and 2 are recruited to these regions, and these in turn recruit or result in certain histone modifications. But later on, we don't need X-cyst anymore. We are going to have DNA methylation and deposition of heterochromatin protein 1 on the X chromosome, and we are going to maintain it in this silent state and then we don't need exist anymore. So this phase is called exist independent maintenance of X inactivation, whereas the initial part is called exist dependent initiation of silencing. Now, yes.
Ajum, can you repeat the part where the maintenance, what happens basically in maintenance?
So we have DNA methylation. So DNA CPG islands are mostly methylated of all genes or most genes. And we have heterochromatin protein 1 condensation or, localization onto the inactive X chromosome. What do you expect if HP1 localizes to one of these X chromosomes? Do you think the inactive X chromosome is going to have a different shape compared to the active X chromosome? What would you think? If we looked under the microscope, would we be able to see a difference between the shapes of these two X chromosomes when one is inactivated and maintained by HP1 interaction? What do you say? Yes.
I think,
yeah, go ahead. But what type of difference do you expect to see?
because the chromatin will be so condensed that we can't see it. We will see it more than the other chromosome.
So that's how X chromosome inactivation was first identified, in fact. In Barr body, you have heard that probably. This is what the Barr body is. Dr. Barr and his colleague in 1949, they looked under the microscope to cat epithelial cells. And this is a cat epithelial cell. They stained it probably with some sort of stain. And here we see the cytoplasm. This is the nucleus, and this is the nucleoli. And right next to the nucleoli here, there is a dot. And sometimes it is close to this, sometimes not, but it is only present, they found, in females and not in males. And they didn't know exactly what this was. They called it the Barr body. But eventually they realized that this was the X chromosome based on experiments like this. They are very old experiments. So this is a prophase and a metaphase spread of DNA from a male and a female. Now you might not see dramatic differences, but the paper which, this paper here published this, says that in prophase, this chromosome here is unusually thick and short compared to the others. And this suggests that there is a thickening going on here. Now we know what thickening is, and we know that it is probably HP1 condensation related. Here again, in metaphase, if this is a male, we have the X chromosome, which they could identify. But in females, when we see two X chromosomes, one looks much thinner and lighter than the other, which looks thicker and condensed. And these were the initial evidences that suggested that there was some sort of condensation going on in one of the X chromosomes in females. Then they did some experiments summarized here, which suggested that there was a particular region in the X chromosome responsible for this thickening or heterochromatinization. The white bars here represent the X chromosome, so the P arm and the Q arm. And if there is a striped region here, it means that there is a translocation that's going on in an abnormal situation, like cancer or so. And they have summarized their data here because it's an experiment from 1974. They didn't have very good silencing tools and, as drawing tools, I'm sorry. And therefore the image is rather amateurish. This is what, things looked like back then. So what they are showing here is these are all X chromosomes that still will be inactivated even in the presence of translocated chromosomes. So for example, this X chromosome, although it has lost some of its P arm, it's lost it completely, the P arm, and it's replaced by sequences from chromosome 22Q, we still see inactivation here. So the inactivating region, the quality that inactivates this chromosome must be contained in the Q arm, because even though the P is lost, we can still inactivate it. And then they extended these experiments further so that further parts of the Q arm and maybe here were lost, and they still could see inactivation. But if this region was lost, they couldn't see inactivation. Therefore, they called this the bar body center, because they didn't know exactly that this was inactivation of X chromosome then. But they found out that you needed this particular region if you wanted to have a bar body. Otherwise, you couldn't get it. There was no condensation. Now, these days, of course, or later on, we have much fancier experiments. This is an in situ hybridization experiment of metaphase chromosomes from a female, and we are staining with in situ hybridization probes specific to X-cysts, and they are in red. And as you can see, there's physical association of X-cyst with only one chromosome, and obviously this must be the inactive X chromosome. Then they did particular experiments like these, and they are, of course, interesting experiments. They, in these two that you see on the top, they transfected cells with a construct that expressed X-ist. And then they did in situ hybridization using probes corresponding to this construct. Because this is a stable transfection, they expected this construct to be present in at least some of the chromosomes. So here we have signal coming from this region in chromosome X, this green signal here, because chromosome X naturally should have this sequence. But here there's another chromosome which contains nice green signals, and therefore we can say that this construct has been stably transfected into the cell and into a different chromosome. Now, this is metaphase, but in interphase, in these cells, we can see nicely that X-cyst, which is shown here in green, covers several chromosomes, and it should not cover several chromosomes. It should cover only one, but it covers several chromosomes because we have been able to stably transfect the cells in different chromosomes with our construct. Any questions in these two figures? All right, and then they did experiments, nice experiments, to show that the X-cyst transcript transfection resulted in the silencing of genes that were transfected together with the X-cyst construct. They generated a construct like this, but the only difference was that it contained a LAC-Z gene somewhere in this construct as well. When the construct with X-cyst and LAC-Z was transfected into cells, as shown here, it was observed that in the early embryonic days, LAC-Z was expressed, but as the cells became adult cells or fibroblasts, we would see suppression of LAC-Z, meaning that the genes transfected together with the X-cyst construct would result in the repression of that gene, regardless of where the construct was transfected in the cell. Even if it was transfected into a chromosome which is not X chromosome, doesn't matter, we still see repression of LAC-Z. That was the conclusion. So therefore, X-ist can independently silence any chromosome and will shut down transcription of genes that are transfected together with it into any chromosome. That was the conclusion from these experiments. Any questions here? Then they said, okay, well, you know, transcriptional repression is an epigenetic thing. So let's see if this is indeed the case. So what we are doing here is we are staining several cells, and this is their nuclei, with DAPI, or exist RNA specific probes, and those are these dots here, and then H3 lysine 27 3-methylation probes, and that's how this image is generated. So the histone 3 lysine 27 3-methylation probe is red, and exist RNA probe is green. If you overlap these, you see that the regions which contain exist also contain very strong lysine 27 modifications. What does this mean?
Exist controls this repression by this 3-methylation.
Right, and who methylates lysine 27? Polycomb repressive complexes. Therefore, it looks like from this initial experiment, it was clear and later confirmed many times that exist must be recruiting polycomb repressive complexes to the DNA which it is going to repress. Okay, any questions here?
I have a question for the previous slide. Here, like by aging, this means like when it becomes like when the embryo is growing, we don't see the laxate. So this means it's not being produced or something.
Right, it's being repressed. Originally, it was expressed on day 6, for example, but in day 13, in both the embryonic cells as well as fibroblasts, the gene was repressed.
So it's not related to age, it's just related to the ability of the cell to start producing the...
Right,
yeah?
exactly, that's right.
Thank you.
All right, so we know that polycomb repressive complexes repress transcription by methylating lysine 27 on histone 3, so this made sense. And they checked other modifications, obvious modifications. For example, here we see an image, a metaphase spread, probably DAPI, we see all the chromosomes, and on the right, we see a staining of the same spread, this time with antibodies specific to histone 4 lysine 12 acetylation. Do you see a difference between the two images? No, yes?
Yes, hocam.
Where do you see a difference?
In the bottom arrow, we see that lack of chromosome.
So here there should be a chromosome, and this one in fact, but it's not there. Why don't we see it?
Because it's repressed, and acetylation is a marker for like
Expression.
yes.
So is this a female or a male cell, do you think?
Female.
Because if it had been a male cell, you would have to have the X chromosome active, and it would be staining with histone 4 lysine 12 acetylation antibodies. So therefore, one of these others here must be the other X chromosome. So it is very clear that the X inactivation results in almost the complete elimination of acetylation signals, which are related to transcription activation. And this is an experiment, an old experiment, which shows this nicely. Any questions here? So how about repressive and permissive methylation patterns on histones? For example, on the top, we have lysine 9, histone lysine 9 methylation, staining together with exist staining. The anti-lysine 9 antibody is staining with red, exist is staining with green. And here we see DAPI. And on the bottom, we see this time histone 3, anti-histone 3, lysine 4 methylation antibodies in red. And here we see exist staining, overlap of that. And here we see exist staining in green. Are these expected results or surprising results? How do you interpret these results? So this is the nucleus of a cell. And why is it red like this in different parts? Because these parts all contain a lot of histone 3 lysine 9 methylation modifications. And one region particularly strong here is shown in red. It is very dense. And interestingly, when we stain the same cell with X-cyst in situ hybridization probes, we see that this very dense region contains this green region. So how do you interpret this data?
Maybe because X-cyst is repressing this part of the chromosomes, so that's why we see very high levels of methylation on lysine 9.
Right, so we already know that X-cyst is related to the inactive chromosome, and we now know that this inactive chromosome must contain a lot of lysine 9, methylation lysine 9 on histone 3. Why? Because what is this going to do? If you methylate at lysine 9 on histone 3, the nucleosomes, what's going to be the consequence of this? What happens afterwards if you do this?
Inactivation?
You by recruiting?
Polycomb repressive complex?
No, HP1. Remember HP1
No.
recognizes lysine 9, methylated lysine 9 moieties. So this is how you are going to recruit HP1 protein to this. Okay, so it means that X-cyst probably recruited histone 9 methyltransferases. Who can recruit histone 9 methyltransferases? Polycomb repressive complexes can do that. So that's probably the series of events. It's recruiting polycomb repressive complexes. They are recruiting histone 3 lysine 9 methyltransferases, and those will ultimately recruit histone heterochromatin protein 1. Now on the bottom, we see again X-cyst, and here we see anti-lysine 4, methylated lysine 4 staining. But here it looks mostly black, except for a single dot here. And our X-cyst is located exactly in this black region. Does this make sense?
John, is this methylation activating?
Right. So you are supposed to memorize this. Lysine 4 methylation on histone 3 is an activating modification. So does this make sense?
I think so.
Why?
Because there is X-cyst there, which silences, and there isn't this activating modification, so the end result is silencing.
Right, so we don't expect lysine 4 methylation in an inactive chromosome. But why is there a dot here? A tiny place, a certain region in the inactive X chromosome contains lysine 4 methylation. What region could this be?
The bar body center?
Be more specific. Why do you expect that in the bar body center?
X-cyst must be produced.
That's right.
Yeah.
So you have to have X-cyst transcription ongoing. So it is possible that maybe this tiny spot is the region where X-cyst is transcribed. It's a very likely possibility. And here now we have HP1 staining. HP1 alpha, beta, gamma, there are 3 types. And what we are looking here is this is DAPI, blue. Green is HP1, alpha, beta, gamma. And then we have red. Red is a region corresponding to the X chromosome centromere. So the centromere region should also be active or inactive, depending on the state of the cell. So here on the left, we see male cells, and we see only one red signal, which is supposed to be the case. We have only one X chromosome. On the right, we have two dots, and this one is very nice. One of them is red, and around that red is a darkish area. And here we have a yellow one, and around the yellow, there is a greenish area. How do you explain these two? What are these two? Why is this red and this one is yellow? If the centromere probe does not overlap with HP1, it's going to look red. If it overlaps with HP1, it's going to look yellow. So why is one yellow and the other red?
One is active, the other one is not.
Which one is active?
You said the overlapping one was the green one. So I guess in the other side is the
Exactly.
active.
So if this is active, does it make sense that the area immediately around this dot is darkish compared to this one, which is yellow, greenish?
I don't know.
Well, what is the greenness? It's HP1. So we said this is active. So if it is active,
yes. Yes, it makes sense. Okay,
right?
good.
Well, now look at this one. Look, it is greenish and yellowish, reddish and blackish. Do you see?
Yes.
So same idea here. And even a little bit here, it is very clear here that there is a greenish area here and this is yellow, but this is not very red. But nevertheless, the area around it is a little blackish. Do you see that?
No, we don't.
Okay, well, compare this one to this one.
Yeah, it's clear then.
Then right. So these all suggestively mean that the maintenance of the inactive X chromosome is most likely due to the recruitment of HP1 proteins.
Hocam, is there a reason why they're on the sides?
Yes, that's a good question. The silent DNA generally is either on the periphery of the nucleus or close to the nucleolus. These are the two regions where inactive DNA is generally supposed to be. So you are right that it is. It's probably correct. Of course, it depends on which side you look at the nucleus. But in these particular examples, it's always in the periphery, and that could very well be related to the fact that it is inactive.
Okay.
Can you go over what the colors mean again? I missed them.
So DXZ1 is a centromere probe for X chromosome, and it's red or orange. And HP1 is an antibody, anti-HP1, and it is green. TAPI is blue. So here we see an overlap between green and red signals. The red signal can only come from an X chromosome because, you know, it's an X chromosome specific probe, but HP1 will come from many places because HP1 can occur in many parts of the nucleus, right? So if you see redness that doesn't overlap with greenness much, then it means that this is an X chromosome region which is not associated with HP1 much. Did you follow me?
Yes.
All right. Any other questions? Now, we saw so far that the X chromosome inactivation is associated with certain histone modifications, HP1, polycomb repressive complex recruitment most likely. So what is this going to result in? DNA methylation. So these experiments now are meant to show us or answer whether DNA methylation in the inactive X chromosome is also a reason for its repression? And the answer is obviously yes. The experiment on the left is an old experiment, and it is the digestion of DNA with restriction endonucleases, analysis on STS, agarose gel electrophoresis, and blotted and probed with a given probe. So this is southern analysis. We are going to probe this blot with PGK1 gene, and PGK1 gene is a X chromosome gene, which is inactivated. So we are going to digest our genomic DNA obtained either from male or female, but spleen or kidney, with either one enzyme, MSP1, or HPA2. Have you heard of these two enzymes before?
No.
So Işık Hoca must have mentioned this. MSP1 and HPA2 both digest the same sequence, CCGG. But MSP1 can digest this sequence regardless of whether the CPG is methylated or not. Whereas if the CCGG, the CG, is methylated on the cytosine, HPA won't be able to digest this. Did you remember this now, or is it new information?
Yes, it's in gene.
No, she said it. Okay.
Right. Okay. So now here we are digesting male genomic DNA from spleen with MSP1 probing. We see primarily 2 bands, maybe a weak band. Here MSP1 from female, and we see again primarily 2 and a weak band. Here HSP1, which doesn't digest if the DNA is methylated, we see the same bands in the male tissues. But in the female, in addition to the bands we saw earlier on, we see these bands here. Why do we see these extra bands shown in asterisks? And we see the same thing in kidney. Male with MSP, male with HPA, female with MSP, female with HPA, and additional bands. Why do we see additional bands?
Ajam, can you repeat the CPG part? Which ones are the one that digest with CPG or without?
Both of the enzymes recognize 5' CCGG. But if the CG is methylated, HPA cannot digest it. If it is unmethylated, it can digest it. But MSP doesn't care whether it's methylated or not. It's going to digest any case. And PGK is a gene on the X chromosome, which is inactivated.
But I thought we're supposed to see something that's not cleaved. But we're still seeing like a smear, and it's kind of not expected.
Well, we are seeing larger bands, right? These are running higher up than the bands we saw originally.
Well, true, but it's still cleaved, isn't it?
Right, because this PGK gene might be in a region where it is inactivated by methylation, but nearby there might be CCGGs which are not methylated.
Okay.
But you can say we should expect all CGs in the inactive X chromosome to be methylated. Therefore, we shouldn't see distinct bands like this, but maybe a big glob on the top, for example. You could have said that for at least one of the X chromosomes. But what we see here is that not that, but we see that there are enlarged bands. So it means that not all CPGs are methylated in the female inactive X chromosome, but at least we have probably more methylation. That's our...
So here in the 6th lane, this means that some of them are not methylated. That's why it's digested more.
So you mean like this one, for example?
This is already present in the other ones, but I mean the new ones, the ones that are next to them, they're stars.
Yes, but why is it present here? Why is it not completely disappeared? Yes, we see larger bands, but why do we still see these?
So both of them are not methylated?
Yes, but this is the active X chromosome most likely. And this is the inactive X chromosome. Because
So the inactive is not completely methylated.
the inactive is somewhat methylated, and therefore we see these extra bands. We cannot say, if there are unmethylated CPGs in the inactive X chromosome by looking at the quantity, the amount of the DNA here versus here. Maybe you're partially right, but nevertheless, it's evidence that suggests that methylation is a mechanism which occurs specifically in genes which are in the inactive X chromosome, because the males don't show such signals. Although, it's a bit weak here, and it would be nicer if these bands had been stronger, and then we wouldn't see anything. But both in spleen as well as in kidney, in the male tissues, we do not see these extra bands. So these extra bands are very likely to be produced from the inactive X chromosome. On the right, we do another experiment, basically the same thing, but now we are digesting with another enzyme called XHO1. And XHO1 is also sensitive to methylation in its digestion site. And the first site that it digests is here, and then there are possible sites 2, 3, 4, and for some reason not 5, but 6. And then this one, which is always digested. Then we digest DNA using XHO1 from kidney, spleen, liver, male and female, and probe using this probe here. If this probe binds to this sequence here, then we see a band here. I don't know what this is, but if it binds to this band, then we see a band here. If this one here, this one here. So if we look at the male tissues, they all have only this band here, because site number 2 is always unmethylated. But if we look at the females, we see this band in addition to this one and this one. So it's a possibility, if we see this one, number 2, 3 needs to be methylated. If we need this, 2, 3, 4 need to be methylated in order to be able to see this band. Are you all with me?
Yes.
So therefore, this means that in females, there are more sequences here corresponding to the PGK1B gene, which is also on X chromosome, that are methylated in most likely inactive X chromosome, because this is only present in the females. The females have a active X chromosome too, and therefore we see this band, but they also have methylated DNA corresponding to the regions of PGK gene, which means that, and because this only happens in females, it means that most likely methylation is also part of the inactivating events in X chromosome inactivation. Okay?
So this enzyme XHO1, it digests the methylated parts.
No, unmethylated. It only.
But then how can we see parts here in the females?
So this band is only possible if you have methylated site number 2, 3, and 4, and therefore XHO couldn't digest those sites, but it only digested this one and this one.
Wow.
You understand? That's the reason. All right. So I think I'm going to skip this. There are some genes that escape chromosome X inactivation, and this is what the evidence for that is, but I'm going to skip this because we don't have enough time. So what is the underlying mechanism that inactivates the X chromosome? We said X-ist is the RNA, and that is produced from the X inactivation center. But actually, the X inactivation center is a much more complex region, shown here. It contains genes that are non-coding RNAs like X-ist, T6, JPX, FTX, as well as some protein coding regions, and some genes which are not relevant to X chromosome inactivation. This one is both a protein as well as a non-coding RNA. Now, the arrows here are important. Exist is transcribed if JPX, FTX, and RNF12 are present. These are positive influences on exist. However, there are negative influences. The biggest negative influences is T6. If it is transcribed, it is going to inhibit exist. And you can see how it might inhibit it, because it's even a larger non-coding RNA than exist, and it's in the opposite orientation. So if you transcribe a lot of this, it's going to pair with exist and degrade it most likely, or maybe even prevent polymerization. And therefore, this is important. TSX and this one here, X site, are positive activators of T6 transcription. T6 does not mean anything. It is exist written in the opposite orientation. So X chromosome inactivation specific transcript, X chromosome inactivation specific transcript in the opposite orientation, right? So there is a lot going on here. And we are going to see a little bit as to how these interplay with each other in chromosome X inactivation. Now,
Artem, which one has the negative impact on the exist?
So you can see here, look, this arrow, hammerhead, is a negative impact. So T6 has a negative impact on exist transcription.
But didn't you say that T6 is the same as exist? Well,
In the opposite direction. It's transcribed in the opposite direction, as you can see here. Therefore, if you transcribe T6, its RNA would be complementary to exist, and that RNA would be degraded.
Okay, thank you.
Now here on the top, it says pairing region. What does pair mean? So pairing, remember we mentioned earlier, well, I'll come to that, but whoa, this is not the slides which I was supposed to show you. I'm going to come back to this. But in X chromosome inactivation, initially we have two active X chromosomes. Then they touch each other. We called this chromosome kissing or touching. And then they separate. One becomes inactive, one becomes active. So this here, these regions here are the regions where the two X chromosomes touch each other. Therefore, they are called pairing regions. We are going to come back to them. But what controls the transcriptional activation of exist and the other genes here? So one very important factor that controls the expression of these non-coding RNAs are these proteins shown here, NANOG, OCT4, SOX2. What are these proteins? What do you call these proteins? What are they?
Maternal effect genes? Or?
No, these are, you remember, if you transfect cells with OCT4, SOX2, and NANOG, you can make those cells embryonic cells. These are pluripotency factors, and they are all transcription factors. These are also pluripotency factors, same here, OCT4, SOX2, KLF4, and as well as these two. What are they doing? So these are produced when the embryo is pluripotent. So in epiblasts. Here we have both the parental and the maternal X chromosome active. Why? Because we are producing a lot of pluripotency factor transcription factors, and they are transcriptional activators of T6. They are transcriptional inhibitors of exist. So if you have a lot of pluripotency proteins around, you are going to inhibit exist transcription, but you are going to activate T6 transcription. What will happen then? Well, T6 will be transcribed, X-cyst will not be transcribed, and you can't inactivate X chromosomes. Therefore, they are all active. Sorry, I'll skip this. But I think, all right. Now, when these two, I'm sorry, let's go over this. Now, when these two chromosomes before inactivation eventually touch each other, then separate, and one becomes inactive. This is what goes on. This pairing region comes together. Here we see the pairing regions coming together. These are both active X chromosomes. And when they come together, the proteins which are sticking onto the two X chromosome sequences touch each other too, obviously. When they touch each other, something interesting happens. So these purple and green dots are pluripotency factors, and CTCF is the insulator protein, which we learned in imprinting. It also sits right there. When they touch each other, here we have now two CTCF proteins, and when they separate, all of the pluripotency factors move on to one of these two X chromosomes. It doesn't matter which one. Whatever they move on, will this be an active or an inactive X chromosome? And this one on the top, will it be active or inactive? What do you say? Here they are both active. Here, what's going to happen? Is this going to be active or inactive? On the top, is this going to be active or inactive after touching and separating?
The top one will not be active because the pluripotent transcription factors are not present on it. So.
Right, and it's going to be able to eventually transcribe X-cyst. X-cyst transcription is going to recruit polycomb repressive complexes, histone modifications, eventually HP1 and methylation, and this is going to be silent. This is going to remain active. So that's how this happens. Once they touch each other, one decides to be inactive by allowing all its pluripotency factors to move on to the other one, and eventually it's silenced while the other one remains active. This is mechanism number one. Mechanism number two is the amount of RNF12 protein. Now remember, in this figure you can see RNF12 protein is a positive transcriptional activator of X-cyst. So if you have a lot of RNF12 protein around, you are going to transcriptionally activate X-cyst. But in the males, you have only one RNF12 gene, whereas in the female, you have two RNF12 genes. So in females, you have obviously more RNF12 in the active X chromosomes. If both chromosomes are active, then you are producing a lot of RNF12. And it can activate or whatever, inactivate something. But there is a critical concentration of RNF12 protein above which it can inactivate X-cyst. Below that concentration, it cannot activate X-cyst transcriptional activation. Because in males, there's only one copy of this gene, it never produces enough RNF12 protein to activate X-cyst. But in the female, because you are producing twice the amount of protein from both active X chromosomes, you have the chance of activating X-cyst transcription in at least one of them. Doesn't matter which one. But the moment you activate X-cyst transcription, X-cyst RNA will silence the RNF12 gene, and you are going to reduce your quantities of RNF12, and X-cyst transcription will stop. But because X-cyst has recruited polycomb and other proteins to the inactive X chromosome, you don't need to continuously transcribe X-cyst. It is going to maintain one of the X chromosomes in an inactive state. This is the second mechanism behind X chromosome inactivation. Any questions here?
Hocam, is it random which chromosome is exactly
Right. I think this makes a difference as well, because as we mentioned here, this cannot be transcribed because the pluripotency factors are sitting here, remember? But this one now is free to be transcribed. I'm not showing you here the active version of it, but to activate this, the RNF proteins can bind to this one and activate it. Even if they bound this one, they will not be able to activate it because these factors are preventing it from being activated, both because they have direct effects, both because they have direct effects in repressing X-cyst, as well as because they can maintain T6 transcription, okay? So your chances of, activating X-cyst here are very low because this one is most likely bound with pluripotency factors in the same time. So even if you had a lot of RNF12 around, it is probably not going to be able to activate this transcript. You are with me?
Yes, hocam.
Any questions here?
Hocam, can we stop because this is too much information for today?
All right, but we have only one mechanism left, and then we will stop anyway. So it's not going to take more than 5 minutes.
All right then.
And I'm recording this, so I'm going to put it up on Moodle so you can listen to it. Because next class we have to finish systems biology, and then we have one more class left for a presentation. All right, so that was the second mechanism. There's a third mechanism, and this relates to how X chromosome inactivation can spread. So this is a complex image, but what it's showing is a simple thing. Here we have no X-cyst transcribed. Then we have the RNF proteins recruit to one of the chromosomes. X-cyst begins to be transcribed. Here we have X-cyst. X-cyst protein, RNA, is shown here to bind polycomb repressive complex 2. Remember, polycomb repressive complex 2 binds to hot air and bunch of other RNAs. It can also bind to X-cyst. So if X-cyst is attached to this chromosome here somehow, it is going to allow polycomb repressive complex to bind to the chromosome. Now I'm going to skip this one, but come to, this one only. So the protein which allows X-cyst to stick to the inactive X chromosome is called YY1. This YY1 protein recognizes a particular sequence in the nucleation center, which is close to X-cyst DNA, and it binds at a particular sequence of X-cyst RNA also. So X-cyst RNA is not allowed to float around in this nucleus. It has to stick to its own DNA by YY1 protein. And because it also contains polycomb repressive complex 2 protein here, this is going to touch different parts of the X chromosome and thereby initiate inactivation. That was the last slide. Do you have any questions here?
So YY1 recognizes what specifically on the X-cyst DNA? Like by X-cyst DNA, what do you mean by X-cyst
So
DNA?
X-cyst, it recognizes the sequence from where X-cyst DNA is transcribed. This is the X-cyst gene here shown with different repeats, etc. There's a particular sequence here to which the YY proteins bind to, the YY1 proteins bind to. And these proteins bind to the sequence in X-cyst, which is called the repeat C sequence, which is made from this region here. So as you make X-cyst RNA from this gene, because there's the protein here, the X-cyst RNA is going to remain bound to the DNA via the repeat C sequence on the RNA. Therefore, X-cyst RNA, although pretty long, is not going to be able to go to other chromosomes. It is attached to chromosome X. And because it also binds polycomb repressive complex 2 at a sequence here called repeat sequence A, imagine that this is bound to right and left, etc. It's going to touch different parts of the X chromosome and start inactivating those parts of the X chromosome because the polycomb repressive complex is going to recruit histone lysine 4 demethylases, histone lysine 9 methyltransferases, and histone deacetylases, and initiate repression. Any questions?
Thank you, Hajj.
All right, so that was what I would like to talk about, and these are not extremely important, so we'll stop right there.
We're not responsible for that, right?
No, you are not responsible for the others, and I'm recording this. I think it's also a video recording, so it will be on Moodle.
Thank you, Hocam.
All the best. I'll see
Have a
you,
good day.
or rather speak to you next week. Thank you, you too.
Have a good day.
Thank you.